Hey guys, welcome to our either third and final or third of a four-part series about Kary Mullis, the alternative AIDS community, and our current events. The focus of this and the next episode, if there is another one, are going to be on the way that Dr. Kary Mullis saw the future of medicine and the things that he had already done before he passed away. As a matter of fact, up to as long as 10 years prior to 2009. And we're going to then look at some similarities between the current medicinal modalities being used against the CV. But first, before we do all of that, I thought it might be prudent to let you guys listen to the man as he's being interviewed for his Nobel Prize. Because I've been saying throughout that he is a particular kind of genius, or he was. And it's this particular kind of genius, I think, that ends up underappreciated in his time, even though a large part of our current science is built on the shoulders of this man, just like with Tesla and the AC electric motor. So try to follow along with him as best you can, but most importantly, listen to what he says about how he came to the discovery of the PCR process.
And I was thinking of this method that would require a couple of oligonucleotides, and one of them would sort of be a control, but they would be pointed right toward each other, just like in a PCR reaction. And then I was thinking of how am I going to get rid of excess deoxynucleotide triphosphate or something like that. There was a couple of technical problems I was trying to solve, and one of them, the way I decided to solve it was to like run a sort of sham reaction first with the oligonucleotides in place and the sample, and then add my radioactive tracers and stuff and run it again, and because it was going to use up it was just not for the idea at first of like duplicating the signal, but then I realized the side effect of that was going to be that I was going to double the signal if there were enough like deoxynucleotides available, and I was trying to get rid of those if there were any coming in with the sample.
No, I haven't stopped there. The thing I've been working on with Charlie there for a couple of years with Charlie, but for about 10 years before that was totally different, and it has to do with trying to come up with a new method. I come up with the method already, but it's making drugs that will be effective against all the bacteria that are now shaking off the hold that our antibiotics had on them. And this new method it's a very empirical kind of thing where you find a site, some kind of an interesting feature on an exterior protein on some bacteria that's giving you a lot of trouble, like say Staphylococcus aureus right now is a good target, and you find a unique thing, something on the outside of that you can model with a little peptide, because it's a part of a protein, and you make a peptide synthetically, and then you select using that peptide. You select from a mixture that contains maybe 10 to the 11th different DNA sequences that you can make now on a machine easily. And you only get one molecule of each one, but you can make about 10 to the 11th sequences. And you now select from those ones that will bind to the site that you have selected on the organism, or to the model of it that a peptide is. And then you do PCR on that one molecule, and you amplify it up to where you have now enough of them to be able to read the sequence of it. And from that, you can synthesize it in large amounts. And now that becomes a specific little label that you can, if you swallowed it, and it was stabilized so that it wouldn't go away in your cells, in your intercellular fluid, if you stabilize it, then that thing will attach itself to, like, say, if you've made it to a protein on Staph aureus, that'll attach to Staph aureus if Staph is in your body. Now, if you chemically attach to the DNA sequence, that kind of DNA sequence is called an aptamer. Now, it has something to do with fitting, because the sequence of that aptamer fits onto some molecule that you've chosen for it to fit. And so if you now attach to that aptamer a little label that is something you're already very immune to, you have a lot of immune like molecules like antibodies to this other molecule. You attach that molecule to this, you've now attached that molecule that you're already immune to something that will grab onto the staph, and so you become immune to the staph. And that's how the system sort of works.
Our approach has been more direct. Can we put messenger RNA directly into cells to create proteins, both endogenous proteins and foreign proteins? And if we could, we believe that messenger RNA could be used to make the full set of proteins in life that might allow us to treat an incredibly broad range of human diseases.
And so we're working out the ramifications. It's not as easy as doing PCR was, because you got to use animals and diseases and people that know about diseases and all that kind of stuff. So we're having a fine time finding collaborators around the world that'll help us do that. But it's working, and it's a simple idea.
Did you have some success with anthrax there?
Yeah, we did. We cured anthrax with some people that we didn't, because I won't come near anthrax. And
Neither
but they
does
had
your
a
wife,
lab
I'm sure.
down at Brooks Air Force Base that we collaborated with these guys that had been working on anthrax for quite a while, and it cured anthrax 100%, which is better than 40%, which is the numbers you get if you use like penicillin, kind of like Cipro is the drug of choice there. But if you get anthrax inhalationally and you know you've got it and you give yourself this drug, you get a 40% chance. And this, with our drug, it's 100%. So it's better. You would choose our drug. And but you're not going to get anthrax. It's just, there's a whole lot of people in the world still that are fighting World War II still. And one of the things that we ended World War II on note was, yeah, we might want to poison our enemies with biological molecules or either with organisms. And we really aren't going to do that. But there's a lot of government money available to do those kind of things, to defend against them. And that's how we ended up with anthrax. I don't see a lot of anthrax, you know. What's that?
Did they fund your research?
Yeah, the research down at Brooks was already being funded. So we just went and I said, why don't you use this method? And we supplied them with the DNA.
But what do you think the future of our ability to basically, get rid of some of these really horrible diseases?
I think it's there. I mean, the idea here is that you really don't need to poison the organism. You just need to reveal it to an immunity that you already have against something else, right? You turn it in, basically. You fool your own immune system in a way, but you're by having this kind of like control at the molecular level of things, you can put your finger in there where you're not supposed to, and you can mess around with it, and you can improve it, because your immune system is amazing that it works at all. But with us directing it, it'll go a lot farther. And things like, there's all kinds of immunity problems that people have, but the biggest one, what I'm trying to approach right now is just, like, if you get exposed to something, a lot of times you don't make an immune response just to everything. So you need to be in the executive seat, in a sense, and say, we want to kill this thing right now, today, and I can make chemicals that are directions for you to do that with. But I need to have that kind of information, the input from, like, knowing this bacteria that you don't like happens to have this structure on the outside of it, and you can use that as a way of pointing that bacteria out to the immune system. The immune system has got a lot of functions. It's got a whole lot of tanks and a lot of serious equipment that's going to waste, because it was made for one disease, and then once that disease is wiped out, it's not useful anymore. You can't take an immune response and retool it. And that's what this system that we're working on does. It takes immune responses that you already have and that are more or less, they're useful to you, but they can be much more so if you can go in and change the guts of them, in a sense, to say, take the guns and point them at this thing, Instead of saying the way the immune system works now, if you get some new organism that you're supposed to be immune to, you have to say, now I have to raise a whole new army, and I have to train it, and I have to do, rather than just saying, ship a few of these guys over here and tell them to point their weapons at that thing. That'll make a big change in the way we become immune to things. Because if we don't have to go through the process of becoming immune to every new threat, we just use an immunity that we already have. That'll make a big difference. Because it costs you a lot of energy to make a new immune response, and every time you do, you have collateral damage, because an immune response is not absolutely specific. So we're trying to, there's a whole bunch of different problems that
Are there some kinds of things that it will work better or worse on? Are there some things that are really, really resistant to the new approach, and some that you think are going to be easy?
The things, nothing right now has shown that it's resistant to it. But the thing about being resistant to antibiotics is there's a reason for how come, bacteria learn how to avoid them. And this doesn't have that element, because they haven't learned how to avoid your whole immune system. They just learned how to avoid this one little antibiotic. This is sort of like going back behind the curtain of the immune system and sticking your hands back through and say, okay, now we're going to change some settings. And so it's not something that the bacteria is going to be expecting. You're painting little targets on things that you would like your immune system to take care of. And your immune system is quite capable of doing it. As soon as it figures out that this is a target,
right.
It's
trained.
It's not
100%.
missing any power. It has all the power you could possibly trust some system to have in your body. You wouldn't want to give it any more. You certainly wouldn't want to have the immune system be able to do this by itself. You wouldn't want to have it change an immunity, because as a new immunity is developed, there's all kind of checks and balances to make sure you don't become immune to your own heart or something. Right? So we're going beyond what the immune system is capable of doing and what you would trust your immune system to do. Because we're saying we already know what the structure is of this thing, and we're going to make something that goes to get that, and we'll make the decisions that otherwise were made by sort of like rote, or by trial and error kind of things in the immune system.
Well, this has got to excite you, because there's so many, things like cancer and things like that we end up having poison bodies for, that you think.
Thinking about cancer more than me.
You are.
I like to start with simple shit like bacteria, but it's there. I mean, the immune system is what keeps you from having cancer in the first place.
Yeah.
So it's possible this will open up that whole area. It's a weird thing that we're doing. We're dealing with that still today. It's getting government grants, right? And so it's easiest if you can explain it to them in the sense of like, some horrible country might use this against us, So don't you want to be prepared?
So that argument is much better than, solving, all
it's much
disease
better than
in the
saying,
world.
yeah, there's a whole bunch of suffering people in the world. Well, screw them. I mean, we're just worried about whether they're going to drop stuff on Manhattan, and what are we going to do about it? And fortunately, if you convince them, you don't have to. The military is totally convinced that some big country is going to drop like bacteria on us. So they're willing to fund that kind of thing, But really, I mean, the purpose for that is to keep people from having all kind of horrible diseases. I would thought it'd be real easy to get funding. It takes somebody like Charlie coming across from London to say, okay, we'll put a few bucks on this thing. But not everybody. I mean, we spent a long time with this idea, probably 10 years, I guess. I don't need any more. But I mean, we spent a long time trying to get somebody to fund it, and always getting money from, from DARPA. They were willing to put up money at first. But they, again, they have a military angle.
So you spoke about this idea of painting targets on bacteria. Would that work for viruses as well?
Yeah, it works for viruses. We're one of the organisms. We were actually getting some pretty good data with influenza until our influenza lady moved to Minnesota. So while she's shut down, we're looking for another influenza person. But yeah, it'll work for anything and the immune system deals with viruses. It deals with things like say influenza, when it causes your cells to start making influenza viruses, it puts a lot of things on the surfaces of those cells that wouldn't normally be there, that the virus wants to put there because it makes it easier for it to do its stuff inside the cell. That's what you use as a target. You say any cell that's been co-opted by influenza is going to have these little things called M2E, which is a little target that we can simulate with like a nonapeptide. So if we because it makes these little holes in cells, and in order to do that, it has to put some of its proteins on there, and they're therefore susceptible to the immune system. So we just redirect an immunity to eat those cells. The immune system's a whole bunch of hungry cells, basically, that are not allowed to eat anything they find. They're only allowed to eat it if it's got a bunch of antibodies on it. That means this is for eating purposes here, and so that keeps them from eating other things. So we're putting those antibodies on there because we find out from the chemists what the flu puts on there, what's different about those cells now. So you don't actually kill the virus itself, you actually kill the cells that makes the virus.
So after listening to all of Kary Mullis's predictions about the future of medicine and listening to what he has actually been doing for the past 10 years as of the recording of that Google Talk at 2009, I'd like to just read you a quick write-up by techart.com about the new Moderna and Pfizer jabs. Unlike more traditional methods of using dead particles or attenuated viruses, the mRNA jab does not come with the parts necessary to trigger the immune response. Instead, the Pfizer and Moderna mRNA jabs contain mRNA strands, literally coding instructions for our cells. They are used by our cells to create a protein antigen. Think of it as a flag that tells our body how to identify a particular enemy. It is the antigen, this flag, that triggers the immune response that teaches our body how to fight the real disease. Pfizer, BioNTech, and Moderna have RNA-based CV jabs that have cleared stage 3 trials with 95% efficacy rates. So this is obviously a previously written article. Both rely on mRNA instructions that code for the SARS-CoV spike protein. So just like Kary Mullis was explaining, you find a particular protein on whatever it is that you want to target, something specific to it, the spike protein in the case of SARS-CoV-2, and you identify that spot, and then you want to flag it with an antigen to make it attacked by the immune system. The mRNA strands in the Pfizer and Moderna CV jabs are used by ribosomes in our cells to create the spike proteins, then discarded. The spike protein flag is presented on the surface of the cell, triggering the body's immune response. Think of it as using your cells to create and wave the enemy's flag. Our body then learns to create antibodies that target the spike protein. If we actually get infected by the SARS-CoV-2, those antibodies will immediately attach to the virus particles, preventing them from infecting our cells, cause the virus particles to stick together, making them easier targets, identifying the virus particles as targets, phagogenic cells to destroy. The immune response to the spike proteins will also train memory cells so that they can respond to a future CV infection weeks or months after the jab.
Scientists have identified a coronavirus protein, the spike protein, that the virus uses to attack our cells. If one could train the body to identify and block the virus-spike protein interaction or to recognize a virus-infected cell, it may be possible to help protect an individual from infection or disease. mRNA vaccines are being designed to instruct your body's cells to make a spike protein, also called the antigen, which could potentially train your immune system to recognize and destroy the virus without exposing you to the real thing. Your immune system recognizes this antigen as a sign of a foreign invader and summons its defenses, like antibodies and T cells. When the real virus comes along, scientists believe that the immune system could recognize the virus's spike and prepare to defend against infection and illness. Because a vaccinated person may not encounter the virus for some time after vaccination, the goal is to train memory cells so that they can quickly respond to a potential invasion.
I'm going to go ahead and say it does. But here's the thing. Everything that Kary Mullis was saying about sticking his finger down in the DNA material and kind of telling the immune system what it should do, he said instead of it doing it rote. Well, no, what that means is instead of doing it the way God designed it to be done. And they advertise all through this Pfizer BioNTech commercial that the mRNA instructions that they're sending into your body cannot make it into your cells, into your DNA. And so they therefore do not change your DNA under any circumstances. That's the claim. But it's very interesting that when Kary Mullis is describing this technology way before we started hearing about it from Moderna, he was saying, but there's got to be a lot of checks and balances to make sure you don't end up immune to your own heart. And I assume what he's saying is if you identify something like a spike protein on a CV, and you think that that's something good to train your body to attack, there better not be any other spike protein-esque things in any of your cells in your body that actually matter. But what do you guys think about this? It's the most bizarre thing. Not only the PCR test is the basis for all of these biologics and the growth in this type of medical field, but Kary Mullis was talking about this and literally being attributed with curing anthrax back in 2009 with this same technology. So now that he's passed in August of 2019 from pneumonia, they're doing another CV situation just like the HIV situation that he spoke out so greatly about. And they're using the technology that he was working on before he passed to cure supposedly this threat. And tell me he's not the Nikola Tesla of our time. All right, guys. Like, share, sub to the channel. Peace.
Thanks for coming here. 10 or 15 years ago, you had some views on HIV and AIDS that were very controversial. So
They still are, yeah.
Where do you stand on
Well,
that now?
what I've come to the conclusion over a long period of time, and a lot more study and reading than most people would sort of give me credit for, because they think, get out of my field, They think it's their field, and that nobody else could possibly have anything to do about it. It's occurred to me, and I think the retroviruses are going to end up a lot of times being like this. The thing that causes AIDS is not a species of the retroviridae, it's the whole genus. And it's the way that there's some members of the genus that are much more effective at doing it than others, but it isn't caused by one, like HIV-1. That was that somebody sequenced that, and then they found, hey, every time we find this, every other patient is a slightly different sequence. And that never struck home in people like Bob Gallow's mind, is that we're not dealing with a species here. We're dealing with, and if you look at the properties of the retroviridae, the genus to which the HIV virus belongs, the way it works is it goes in and it's a DNA thing. It like sticks its DNA into lymphocytes, and the DNA just sits in there until that lymphocyte starts to divide itself rapidly. And then the DNA starts making protein, or RNA and then protein, and then making particles that are infectious. And it's a cyclic kind of a phenomena. It goes around, now it infects new cells and it starts doing that. But it doesn't immediately start doing anything. It goes into the cell and waits until that cell starts to divide rapidly. So what that translates to in the immune system is that once a sequence, because those sequences mutate really fast, because reverse transcriptase is involved in their replication, and that makes mistakes at 100,000 times more often than like DNA polymerase does. So there's a real, that genus is very, very heterogeneous inside of it. And what's happening is you get a sequence that starts replicating itself. It makes some mistakes. It infects other cells. It sits there and waits. Most of the time, it's just a piece of DNA. It's ridiculous to fight it only at the time when it's proteins and it's infectious particles. That's what they do. I mean, all the retroviral drugs can only be direct. They only are effective against the RNA containing protein form of the disease, and they can only just do so much. They'll modify the rate at which it happens, but the real problem is that you've got lymphocytes that have all these sequences in them that are sitting there waiting to their good time. When the lymphocyte starts reproducing itself rapidly, they reproduce. When it's not, they don't. So you can't kill it by stopping it at the protein level. You can kill its action. You can stop it from taking action right now, but it's still got the DNA sequence in a bunch of lymphocytes. So until we figure out how to deal with that problem, I don't think we'll have a useful drug against any of the retroviral. They're coming up with new ones. There's new things that have been blamed on retroviruses. And the very first thing that people have to recognize when dealing with them, that they're dealing with they're not just dealing with one single thing. Like one sequence is not, just because you can only detect that one. See that, there aren't any nice tests for retrovirity in general. There aren't any tests. So you could give them to people and say, my God, people have got all kinds of retroviruses. But they only have a very specific test for the proteins that are involved in HIV. So they can tell you whether you got that, but they can't tell you whether you got a whole lot of related things. And that's really the difference between people that get sick and people that don't, is that people that get sick have a whole lot of different versions of the whole thing. This is my feeling about it, and it's not an unsupported kind of feeling. It's from just looking at the data. More and more data is coming forward that supports that idea that it's not just HIV as originally defined by Bob Gallo and Luc Montagnier. It's a whole bunch of things, and they're retroviral, but they don't have the same sequence. And just killing one particular little cluster of those guys is not going to stop the disease.